Right, welcome to, I don't know how many of these streams we've done, but welcome to a biology revision stream. I want to clarify, and I'm going to have to do this at the start of every stream now, because every time I do a biology stream, I get at least 1 or 2 subscribers. Guys, I am not a teacher, I am not qualified, I don't know what I'm doing, okay? Please don't watch these streams as an alternative to revision, or even as revision, okay? What I'm doing in these streams is literally going over content that I don't know too well, things like that. Please don't watch these streams as a method of revision. That's not the purpose. The purpose is for me to revise using these streams. There's better people out there. Right, okay. We're not opening the realm. So, what I want to do today is firstly I want to go over the entire spec for paper 1 and see what exactly I need to work on and what I don't need to work on, and then we'll just go from there. We'll look at some paper questions. I can probably stream for about an hour, so my voice is going to be in tatters by the end of this, but that's fine by me. Yeah, okay, we'll go over further maths. So, biological molecules. Now, we have already revised, in full, monomers and polymers, carbohydrates, ATP, and inorganic ions. We are yet to go over lipids, proteins and enzymes, nucleic acids, and water. That could be a decent, quick thing to go over today, especially water, because there are a lot of people saying that this year the paper 3 question is going to be on, why have you wrote insects on the board? In relation to mass transport of insect. Why the fuck is everyone watching this stream? Guys, this is revision for biology. So water is supposed to be a predicted essay question this year, which would be nice. You could talk about it in relation to transpiration, translocation. You could talk about it in relation to its properties of having a high heat capacity, therefore preventing the denaturing of enzymes. You could talk about water acting as hydrolysis, condensation, the metabolite. So we'll go over water today. In terms of cells, we have cell structure and microscopy. That would be a nice one to go over. Cell cycle and mitosis, we've gone over in class. I think we could do with a quick look over. Transport across membranes, we probably don't need to do much on. The only things I don't know for that topic is explaining the fluid mosaic model. I'm going to turn off chat. Sorry, guys. The one thing that I don't know is explaining the fluid mosaic model. And the second thing I don't know is how to explain the interactions in the the saturated and unsaturated fatty acids, the hydrophilic, hydrophobic regions. What the hell am I on about? No, it's not unsaturated and saturated. It's the fatty acid tail, the glycogen head. That's what I'm on about. My god. And then the immune system, which we should definitely go over. Then, in terms of everything else, we have surface area to volume ratio. We need to go over that, definitely. Gas exchange, I don't know whether we should. I don't see... Probably for insects and fish we'll look over, yeah. Digestion and absorption, 1 or 2 questions wouldn't hurt. Mass transport in animals, maybe. Mass transport in plants, yes, we'll go over. Then finally, genes and chromosomes, I can't see what harm it would do going over that. Transcription and translation, probably not as much. Mutation and meiosis, we are saving right up until exam starts. Genetic diversity and adaptation, I'll happily go over. Species and taxonomy, I'll happily go over. Biodiversity, I'll go over. And investigating diversity, that's things like quadrats. I don't see a reason to go over that. So, We will start off by going through the very first thing in the spec, which is water. And from there, we will just kind of crack on and see what we can do every single day up until the exam. This is a 5-mark question. So let's go over this. And this question seems to be a framework for other answers, which is good. Just making sure. Okay. Explain 5 properties that make water important for organisms. I can't even explain 4. Okay. Let's think about this logically. The properties of water. Number 1, high specific heat capacity. It wants it to important. So this is basically like the essay question, just shortened. It means a high amount of energy is required to raise the temperature of water. The importance of that is it allows it to act as a buffer. So, obviously, realistically, things like the ocean and the sun, like, the ocean is able to maintain a stable temperature, primarily because it's a lot of water. Like, that's common sense. And the water acts as a buffer. Next, something about cohesion and the column, I would say. Properties of water. One obvious one would be it acts as a metabolite for hydrolysis and condensation. And if you really wanted to go into detail, you would say hydrolysis is the addition of a water molecule to break apart bonds between monomers. Condensation joins together bonds between monomers. So that's 2 properties. So we've got at least 2 marks. How are we going to pull this off? Strong cohesion tension. Strong cohesion tension means that water moves in a column, and that is due to the interactions between the H bonds. Then we would say water is a solvent. Therefore, it allows substances to be dissolved in it. The final one, I don't know any other properties of water. High specific heat capacity, strong cohesion tension, it's a solvent, it's a metabolite. I'm missing an obvious one. I know I am. Something to do with latent heat, I remember that, but I don't know how you would explain that. What other properties of water? Polarized, would that be one? Because the oxygen is negative, the hydrogen is positive. You know what, we'll just put it. It's a 5-mark question. What harm is 1 mark going to do? Oxygen is negative, hydrogen is positive. Right. Cool. Question answers. A metabolite in condensation, hydrolysis, photosynthesis, or respiration. Cool. 1 mark. A solvent. Okay. Now it's telling me here, the explanation of that is allowing transport of substances or allowing metabolic reactions. I explained what a solvent means. So let's rewire our definition here. A solvent allows the transport of substances. Close enough though. I would happily give myself 1 mark for that still. High specific heat capacity, so buffers changes in temperature. Yep. Cohesion between water molecules, so supports columns of water. So produces surface tension, supporting small organisms. Okay, let's just take that because, yeah, it allows water to move in a column or supports columns of water. Yeah. The other property is latent heat of vaporization. I would not have known this. One thing I want to do on my revision document now, and I should probably write this down so I don't forget, is I want to make a document quickly on the properties of starch, cellulose, glycogen, ATP, DNA, mRNA, tRNA, water, because you could get asked a 5 or 6 mark question on any of those. A question saying, compare and contrast the properties of starch and cellulose for 6 marks. That's appeared before. 4 mark question on contrasting the structure of DNA and tRNA. Question right there that we're doing now, a 5 mark question on properties of water. Best case scenario, I got 4 marks, worst case 3 marks. Latent heat of vaporization provides a cooling effect through evaporation, so that's a nice one. That is a very nice one to remember. Very nice one to remember. Okay. Solvent allows the transport of substances. That's easy to remember, just double S. Solvent, substances. Yeah, starts with an S for substances, ends with a T for transport. That's so gimmicky that it might work. Oh, hello. I don't know how long you've been watching. My apologies, man. My apologies. I had chat turned off because everyone in my nans apparently were watching. I don't know if you're still here, but we're going over the properties of water. So this is still the biological molecules topic. I was just quickly talking about how I want to do some revision on that. Oh, okay. I was just talking about how I wanted to do revision on the properties of all those molecules. Because in paper 1, you will absolutely get asked a question at some point on, I don't know, a 4 mark, explain the properties of ATP, contrast the structure of whatever and whatever. The nicest one you could get would be contrasting the structure of ATP and DNA. That would be the nicest set of marks in your life, because they are basically, the same. They are basically the same molecule. I probably pissed off a lot of people saying that, but it's true. You have the adenine base, the phosphate ion. Sorry, I've done this the wrong way around. Phosphate ion, phosphate, and the ribose sugar. Then in DNA, instead of a ribose sugar, it's deoxyribose. There's 1 phosphate ion instead of 3, sorry. And then there is a base. So the literal only difference is ATP has ribose, DNA has deoxyribose, ATP has 3 phosphate ions, DNA has 1 phosphate ion, and ATP always has the adenine base. DNA has 1 of 4 bases: adenine, thymine, cytosine, guanine. ATCG. Okay, I'll open the realm right now. So, properties of water. We have that nailed now. I will happily add some stuff to revision document later. Best case scenario out of 5 marks, I just got 4. Worst case scenario, I got 3 because I didn't explain what a solvent is properly. Next question. Another question on properties of water. So it looks like if this were to appear in an exam, it would likely be explaining a property, which means this is going to be significant for revision. State and explain the property of water that helps prevent temperature increase in a cell. Obviously, that's going to be high specific heat capacity, allowing it to act as a buffer solution because a large amount of energy is required to raise the temperature of water. High specific heat capacity, 1 mark. Buffers changes in temperature. Accept ideas such as a lot of energy needed to change temperature. See ya, 2 marks. And what? Right, that is a repeat. Explain 5 properties that make water important for organisms. That is a repeat. There is no way they've asked that twice. Each for 5 marks. State and explain the property of water that can help, but what the why are there so many of the same question? State and explain the property of water that can help buffer changes in temperature. For 2 marks, but it's worded differently. It's the exact same answer. High specific heat capacity. A large amount of energy is required to raise the temperature of water. Therefore, it acts as a buffer. That is an insane thing. The fact that every time they've asked a question about water, it's either been that property or just listing all of them. That is crazy. Water and inorganic ions have important biological functions. Give 2 properties of water that are important in the cytoplasm of cells. For each property, explain its importance. Okay. 4 marks. 4 marks for picking out 2 of these. What the fuck? Why are the water questions so nice? That's kind of weird. Most obvious one, high specific heat capacity. I've literally just said that 10 times. That would be 2 marks. Property 2, you could pick any of these. I would say the most obvious one would be in the cytoplasm of cells, water acting as a metabolite, allowing for hydrolysis and condensation reactions, but also obviously water acting as... No, they don't, man. That is the thing. Question... Mark scheme. Oh, okay, wait, you might be onto something here. This mark scheme has like 1 word per sentence here. First one for saying, wait, hold on. Right, this mark scheme is dodgy, but like, but it's written wrong. The question is saying name the property and then explain why it's important. All it's doing is naming the property. But the mark scheme's literally clipped off. Yeah, there's a mark scheme. There's a point for saying that it takes place in hydrolysis or condensation reactions. There is no mark point for saying anything about heat capacity, which is weird. Because that would be one. Because otherwise, if the temperature wasn't buffered, enzymes would be denatured. There would be no reactions occurring at all. That is a dodgy mark scheme. They have formatted that incredibly wrong. Apparently you could get a mark just for saying reactive. Literally, like, a mark for saying the word reactive. That is crazy. I feel like it's thing is, I'm not using the AQA mark schemes. I'm using Physics Math Tutor. I don't know where they source their questions from. I know it's AQA, but they definitely obviously modify the mark schemes, given how it's literally like that, like it's just written like that. So I'm willing to go out on a limb there, and saying that I would probably still get 4 marks for that, even though according to this mark scheme I would have only gotten 2 marks. Because yeah, heat capacity is absolutely a property of water. It's in the cytoplasm. Otherwise, the properties are similar. It's talking about it being a solvent, talking about it acting, and it being a polar molecule is one, which I mentioned right at the start of the stream. And that is it. That is everything. That is every question on water. That is crazy. So the only questions they have ever asked about water, except obviously in the transpiration topic, the only questions they've ever asked about water is this property here, and then like literally just picking out any of those other properties. Oh yeah, of course. Thank you, man. Take care. Thank you for watching. Let's move on then to nucleic acids. So we just spent 20 minutes going over water. That's crazy. And my Wi-Fi's gone down. What the fuck? You know the function. I'll just go grab a drink while I'm waiting for that. Man. I'll turn off chat again just so I can concentrate for like right. I see. 2 marks. So this is DNA. So let's get rid of this. I'll just stand back to take a screenshot and then. So I'm happy that we've gone over water because I would not have remembered anything about latent heat. I wouldn't have remembered anything about what a solvent is. Solvent, I'm going to remember that now. Solvent, S to T, substances transported. Easiest thing ever. Right. DNA, deoxyribose nucleic acid. We will draw it, first of all. So it's phosphate ion, ribose sugar. I don't know what comes first, actually. But that is roughly, I hope, structure of DNA, like a single strand. I can't draw backwards. Guys. You're gonna have to just trust me. I'll even Google it quickly. DNA does come up significantly in the paper 2 when talking about gene technologies. I would not bother revising that just yet. Because let me be real, that is such a bad topic. Right, yeah, I think I've drawn it right. Okay. So, it's saying, name the type of bond between complementary base pairs. Complementary base pairs, it is hydrogen bonds. And between adjacent nucleotides, that's fancy wording of saying this bond here, it is a phosphor diester bond. Cool. The length of a gene is described as the number of nucleotide base pairs. Use information in the above diagram to calculate the length of a gene containing 4,380 base pairs. And it wants it in nanometers. I would not know how to do this. Let's try this math question, though. Let's give it a go. So it's saying 4380, I think, if I've converted to standard form correctly. Have I converted to standard? Oh my god. Well, I'm going to get a calculator in the exam. I don't know why I'm stressing about this. Where is my calculator? Oh, right there. Okay, let's try this. 4.38 times 10 to the power of 3. Yeah, okay, I was. So that's how many base pairs? Now the diagram is showing that in 1, 2, 3, 4, 5 base pairs, it's 1.7. Therefore, we can divide 1.7 by 5 to get 0.34 nanometers per base pair. Hope I'm doing this right. Then we just multiply that number by that number. Surely I'm doing this right. 4380 times 0. It's a 2-mark question. This can't be bad. And it's saying 1489.2. Let's round that up to 1489. Let's have a look. Oh no! Wait, no, correct answer. Yeah, I got it right. Okay. Yay, I got it right. I did it right. Let's go. I'm happy. I'm actually happy because all the math questions are horrible in biology. I got the simplest one right, but I'm happy. Okay. Yay, we did it. Hell yeah, fuck you. Right. Oh, what? Complete the table below to show which elements are found in the following molecules. What? Okay. Galactose. Phospholipid. RNA. And sucrose. And the elements are carbon, nitrogen, oxygen, and phosphorus. Ooh, for 2 marks! For 2 marks, that is insane! Right, galactose. Let's think about this logically. We're not rushing this. For 2 marks, we're gonna rush this. Carbon and oxygen, because it's a carbohydrate. Phospholipid. It's carbon chain with hydrogen and it has the pi. I don't know about carbon. I mean oxygen. Because it has a glycerol head. Does that have oxygen in? I don't recall. RNA. RNA definitely has that. Definitely has the nitrogenous base. Would it have all of them because of the sugar? And sucrose, I think, would be the same. How close? Come on. Oh, I was 1 tick off. Phospholipid did have oxygen in. Fuck. Everything else was right though. That is a good piece of knowledge to have though. Phospholipid is literally carbohydrate plus... Man, that was a fun question, you know. That was a fun question because that's nothing that we would have been taught. We would never have been taught a specific atomic structure. Which of these is supported by the results in figure 2? Q, because it's semi-conservative. Explanation for the first unsupported model. First unsupported model shows the second unsupported model here shows DNA being randomly crossed over. It's not supported because the concentration of DNA is not random between generations. And in the second one, the density is not the same. I don't know. Yeah. Model P is unsupported because there should be 2 peaks in generation 1. Model R is unsupported because there should be more than 2 peaks. I would not have understood that. Describe the structure of DNA. Okay, I'm assuming for this we're allowed to draw a diagram. So, describe the structure of DNA. We are going to say that it is deoxyribose sugar plus nitrogenous base plus phosphate ion. It is double stranded. So 2 strands of polynucleotides. I would obviously talk about H bonds join together, nitrogenous bases, phosphodiester bonds join adjacent nucleotides. That will probably be enough marks. Let's have a look. Allow polynucleotide. Allow each nucleotide formed from deoxyribose, phosphate, and organic nitrogenous base. So I said this, but I didn't explain what that was. I guess I would have gotten a mark. Saying it's double-stranded would be 1. Allow phosphodiester bonds, allow hydrogen bonds. Surprised you wouldn't be allowed to draw a diagram there. Describe how a phosphodiester bond is formed between 2 nucleotides. This would just be saying that it is a condensation reaction joining together the deoxyribose with the pi of the inorganic of the phosphate group of the adjacent nucleotide. So the deoxyribose of one nucleotide is bonded together via condensation reaction. That's the important one. Condensation to the phosphate group of the adjacent nucleotide. Condensation between phosphate and deoxyribose, and you have to mention DNA polymerase. I forgot about that, but it was 2 marks, so we still would have got it. That's an important one, is talking about enzymes. I remember there was a question on saying how ATP is formed by its structural molecules. Exact same premise for the question. You would talk about its components, how they would be joined together via condensation, explain what condensation is. Then there was a hidden mark point for explaining the enzyme. In ATP's case, it would be ATPase or ATP synthase. DNA's case, it would be DNA polymerase. So close enough, but yeah. The two strands of a particular gene contained 168 guanine bases between them. And this is a formula. G equals 4A plus T minus C. Is this algebra? Calculate the max number of amino acids and your understanding of DNA structure. So the maximum number of amino acids. Shit. So that's triplets. Okay, let's put that in mind. 168 is equivalent to 4 multiplied by something plus something minus something. What could this even be implying? Two DNA strands contain... So it's two strands between them. Is that important at all? Would that not require us to divide? Would that not require us to divide? If it's saying between two bases... Wait, okay, hold on. Let me do this the way I think it's meant to be done. Complementary base pairing. So no matter what, you are going to get minus 168 because it's the same, complementary. If we add 168 to this side, we get 4A plus T equals... I should have got Declan to help me with this. This is 3 marks, 2. So no, I'm definitely doing this wrong if it's taken me doing algebra for this. 336 equals 4AT. Then I would divide by 4 to get 84. Then times it by 2 to get 168. So overall, is it 168 plus 336? 84 plus 84. Oh, I don't know. I give up. Correct answer is 70. I accept 420 total bases in gene. Oh, I was close. I said that the answer was 336 divided by 4 would be 84. I didn't think of adding them together. I might have gotten a mark. Best case scenario. That was difficult for 2 marks. That required some intense, reverse engineering. For 3 marks, I would understand. And I'm sure for 3 marks, they would give me a mark for just getting a bit of it right. I might head off in 20 minutes, but we'll get fatty acids done as well before I go. Oh my god. Fuck me. What a day. Right. In the process of semi-conservative DNA replication, 2 strands within a DNA molecule are separated. Each then acts as a template for the formation of a new complementary strand. Explain how the separation of strands occurs. Oh, are we allowed to say hydrolysis? Is this one of the mark schemes that allows it or not? Hydrolysis, we'll just put that in brackets for now. Or breaking of H bonds between complementary bases or base pairs catalyzed by DNA helicase. Please be right. DNA helicase breaks hydrogen bonds between base pairs. Reject hydrolysis. Okay, so you are never allowed to say that it's hydrolysis. Good to know. Breaking of hydrogen bonds between complementary base pairs. We know how DNA replication works. I don't need to go over it. I will, just because it's fun. DNA helicase breaks apart the bonds between the hydrogen bonds between the complementary base pairs. So you get left with something like this. You get me? Where one strand acts. No. Both strands act, because this isn't transcription. Both strands act as templates for free-floating nucleotides to bind with their complementary bases, to align with their complementary bases, a bond with them, forming hydrogen bonds. From there, they are catalyzed by DNA polymerase, which catalyzes a condensation reaction between the adjacent nucleotides, forming two strands of DNA, each strand with an original, each double strand containing a strand of the old. Describe the role of DNA polymerase. DNA polymerase joins together adjacent nucleotides via a condensation reaction, forming phosphodiester bonds. Yep, all 3 mark points. Name the 2 scientists who proposed the models of chemical structure of DNA and DNA replication. Miesel and Stahl? Or is it Watson and Crick? It was Watson and Crick. Watson and Crick discovered the structure. That is a nasty mark because, who cares kind of thing. I'm sorry, that's not nice. Scientists replicate DNA in a test tube. To do this, he mixed an enzyme with identical single-stranded DNA fragments and a solution containing DNA nucleotides. Name the enzyme used in this DNA replication. He mixed an enzyme with single-stranded fragments. DNA polymerase? That's a weirdly worded question. Yeah. Use your knowledge of semi-conservative replication to suggest the role of the fragments and the role of the nucleotides. The fragments will act as templates for the base pairing. Template, yep. How many marks is this? 3. I might not get it. The role of DNA nucleotides would be to bind to their complementary base pair and form a second strand, a complementary strand. Forms complementary pairs or forms strand. Okay, 2 out of 3. Draw and label a single nucleotide. We've just done that. Give 2 features of DNA and explain how each one is important for the semi-conservative replication. 2 features of DNA and explain how each one is important. Should we say complementary base pairing? Therefore, when the base pairs are broken, free-floating nucleotides combined with them. Complementary base pairing allows replication. The other one is weak hydrogen bond allows easy separation. That is okay. That would have been one I got. Oh, I need a question on the process, the structure of DNA. Contrast the structure of ATP and a nucleotide found in DNA. We've just done that. Why do the arrows move in opposite directions? Two strands of a DNA molecule separate and each acts as a template for the production of a new strand. Why do the arrows move the other way? Why is this 4 marks? I don't know. Anti-parallel strands. Enzymes have active site. Okay, 1 mark, would have got that. Only substrates with complementary shape combined with the active site. Okay. I would have gotten a mark or 2 for just talking about that. So if you're ever asked a random question that you have 0 idea on, but it relates to enzymes, always mention active site, complementary. 2 marks. I need a question on the structure of DNA, please. Describe a structure of DNA for 5 marks. Let's look at this. We've already done that. I need one of the properties of DNA. Really? There's not a single question. Okay. Well then. I am happy to wrap things up there, to be completely honest. Mainly because I'm a bit tired, but that doesn't mean I'm going to stop. I'm going to now crack on. I will do this by the end of tonight. I will go over the properties of starch, cellulose, glycogen, ATP, DNA, mRNA, tRNA, and water. I will go over at least 3 or 4 properties of each one, so that when it comes to exams, there can be some nice flashcards. But yeah, that's about it for me, guys. I haven't been live for too long, to be honest. But yeah, now that revision is kind of getting a bit, like, the thing is, we have our history paper in exactly a week and a day now. So I'm more beginning to shift my focus over to that. Even though I'd rather not, because I want to revise biology more. But yeah, at the end of the day, all I really need from my A-levels are 2 Cs, which I know I should be able to get. So worst case scenario, do your best, guys, and yeah, take care.